Start this.
Okay, good morning, students. In yesterday's class of protein chemistry, we were discussing about the structural organization of proteins, how the proteins attain their three-dimensional structure by undergoing the 4 levels of organization, and even we have discussed about the denaturation. So that means denaturation means when proteins subjected to various physical and chemical agents, they will undergo denaturation. Denaturation results in the changes in the secondary, tertiary and quaternary structure. Primary structure is intact. Now here, because of the changes in the secondary, tertiary and quaternary, there is a changes observed in the physical and chemical properties. So denatured protein is less soluble when compared to the original protein. Now here, denatured protein results in the denaturation results in the it decreases the solubility of the protein. That would result in the formation of precipitation. But if you remove the denaturating agent, sometimes it may be reversible, but most of the time denaturation is irreversible. Then coming to the next topic, protein folding. Protein folding means already in the previous class we were discussing about, in order to attain the complete functional biological activity of the protein has to undergo folding process. Okay, organization is nothing but the protein folding in the 3-dimensional. So protein folding in 3-dimensional, protein has to be folded in 3 dimensions to become the fully functional or to become biologically active. So protein folding, it is an inherent property which is specific to each protein for its biological activity. And this is done by a specialized protein. Okay, protein folding is done by a specialized protein called chaperones. Chaperones are the proteins which help in the correct folding of the protein. So we know that in order to attain the 3-dimensional, protein has to fold. There are specialized proteins called chaperones which help in the correct folding of the proteins. So chaperones are polypeptide chains. These are the special type of polypeptide chain binding proteins. Okay, these are polypeptide chain binding proteins which help in correct folding of the protein. And here the chaperones which help in assembly of the tertiary and quaternary structure. Now here, and they facilitates these chaperones, they facilitates the proper interaction on the protein to give the correct compact 3-dimensional conformation. How means by stabilizing the intermediates. Okay, these chaperones, they will help in the correct folding of the protein by facilitating the proper interaction of the protein to form the correct folding and compact form and specific 3-dimensional form. And there are different types of chaperones have been identified: heat shock proteins and chaperones. So these are different classifications. Chaperones are classified into heat shock proteins and chaperones. Now here, heat shock proteins are one of the type of chaperones. They are named according to their molecular weight. Okay, examples of heat shock proteins are HSP60, okay, and HSP90. Okay, and this 60 and 90, what does it represent? Means it represents the kilodalton molecular size. Okay, okay, so HSP 60 and 90, this kilodaltons in size. Okay, now here these examples of heat shock proteins are HSP 60 and 90 which are the type of chaperones which help in proper folding of this one. And this 60 and 90 HSP 60 and 90 together called as chaperonenes. Okay, chaperonenes together, this HSP 60 and 90 together called chaperonenes. Now here protein misfolding. So now that till now we have discussed about there are special proteins by name chaperones. Okay, examples are different types of chaperones are heat shock proteins which help in the correct proper folding of the proteins. Now here there is misfolding. Some diseases are related to protein folding. Okay, now here this misfolded proteins are usually tagged and degraded within the cell, and deposits of these misfolded proteins are associated with a number of diseases. Okay, if the proteins are wrongly folded, they will deposit. If they are easily degraded within the cell, and their deposits they may be deposited within the cell and causes a number of diseases. So diseases which are related to misfolding of the proteins are prion diseases, Alzheimer's diseases, and amyloidosis. Okay, so these are the diseases which are related to misfolding of the proteins. So why the proteins will undergo misfolding means if any stress to the cells like free radicals. Okay, if there is any stress to the cell like free radicals, bacteria, heat radiation or toxins which may cause the stress to the cell. So because of that stress, there is increased production of the heat shock proteins. Okay, any stress to the cell increases the production of heat shock proteins. Now this heat shock proteins leads to the increased production of heat shock proteins which causes protein misfolding. Actually, what is the function of heat shock proteins? Help in the proper folding. If there is increased production of this heat shock proteins are there, that would lead to increase that causes the protein misfolding. So protein misfolding that leads to rapid degeneration and results in the accumulation of this degraded ones, accumulation of the aggregation. Okay, that leads to a number of diseases. Okay, so any stress exposing of the cell to the free radicals, bacteria, and heat radiations or toxins which causes the increased production of heat shock proteins, that causes the protein misfolding. Protein misfolding leads to rapid degeneration and results in accumulation of aggregation. Now here these misfolded proteins are called prions. So these misfolded proteins are called prions, and these prions can transmit their misfold shape into normal variants of the same protein. Okay, see here these misfolded proteins, they are called prions, and these prions can transmit their misfold shape into the normal cells. So that causes severe fatal and transmissible neurodegenerative diseases in the humans, mainly damage the brain cells. So these prion accumulation in the brain that causes memory impairment, personality changes, difficulty in movement. Okay, see here the protein misfolded proteins are called prions. When the proteins are misfolded, if they exposed to, if the cell is stressed, that would form the misfolded proteins which are called prions. The property of the prion is they can transmit their misfold shape into the normal variant that causes the severe fatal diseases and causes the transmission of the neurodegenerative diseases, especially brain cells are damaged. That causes memory impairment, personality changes, difficulty in movement. Now here, protein misfolding in humans causes disease. Kuru disease includes and Fels-Jakob disease. And misfolding of the protein causes spongiform encephalopathy in the cows. Now here, there is amyloidosis. Amyloidosis means these are misfolding of the proteins may occur spontaneously or may be caused by a mutation in a particular gene, which then produces a altered protein. Okay, see here the misfolding, sometimes the proteins are misfolding. Misfolding of a protein may be it may occur spontaneously or it may be caused by a mutation in the particular gene, which then produces a altered protein. Okay, see here. Now this amyloidosis means these are the group of diseases caused due to incorrect translation on the ribosome. Now here what we are discussing, the misfolded proteins causes various diseases together called as the associated. The diseases related to misfolded proteins are called amyloidosis. Amyloidosis means these are the group of diseases caused due to incorrect translation on the ribosome. Okay, now here incorrect translation on the ribosome that results in abnormal deposition of an insoluble form of the protein called amyloid. Okay, see here, if there is any incorrect translation on the ribosomes is there, what will happen? That results in abnormal deposition of a protein, insoluble protein called amyloid. Amyloid is a protein which is insoluble. It gets deposited especially in the major organs and affecting their functions, leading to life-threatening organ failure. Okay, see here, this insoluble amyloid, it gets deposited in the major organs, vital organs, and accumulation or deposition of this causes their failure, multi-organ failure, and that causes the life-threatening organ failure. And amyloidosis, mainly the accumulation of this amyloid protein mainly causes Alzheimer's disease, Huntington disease, Parkinsonism disease. Okay, amyloid, what is amyloid? It is an incorrect translation on the ribosome would result in the formation of an insoluble protein called amyloid. If it gets deposited in the organ, it causes organ failure. The diseases, amyloidosis causes like Alzheimer's disease, Huntington disease, Parkinsonism disease, etc. And what is, we all know that Alzheimer's disease, Alzheimer's which is seen in the elderly people, that may be due to misfolded protein of beta amyloid protein in the human brain. Okay, see here, in the aged, okay, elderly people, Alzheimer's disease is there, a misfolded version of the beta amyloid protein in the human brain. Okay, misfolded version of the beta amyloid protein in the human brain that causes the accumulation or deposition of the fibrous proteins or the plaques that causes dementia, progressive loss of the memory, etc. Okay, or there is depression is also there. Okay, Alzheimer's disease, here there is a misfold of the beta amyloid protein in human brain that causes the accumulation of the fibrous deposits or plaques in the brain that results in depression, dementia, progressive loss of memory, etc. Okay, sorry, it is not dementia, this is a maybe depression, progressive loss of memory, dementia is there. Okay, this is Alzheimer's disease. And okay, that's it about this protein folding. So protein folding, chaperones are the proteins which are responsible for normal folding of the protein, like heat shock proteins. And if there is any stress of the cell causes the synthesis of the production of the increased production of the heat shock proteins, that is heat shock proteins and chaperones that causes the production of the which causes the misfolding, increased production of the heat shock proteins, which folding increases the which causes the production misfolding of the protein, which gets deposited and leads to various and that deposition in the major organs causes various diseases. Okay, that's it about the protein folding and misfolding. And then coming to the next one is, next topic is genomics and proteomics. Okay, these are the genomics means it is the study of the entire set of the genes in the genome. Okay, genomics is the both are closely, genomics and proteomics both are closely related fields. Genomics is the study of entire genes in the genome, but proteomics is the study of set of proteins expressed by the genome. Okay, expressed by the genome. So that means study of proteomics is nothing but the study of proteome. Proteome means it is the complete set of the proteins encoded by a particular gene. Okay, so genomics is the study of entire set of the genes in a genome. Okay, proteomics is the study of proteins expressed by the genome. So genome means that includes the complete set of the proteins encoded by the particular genome. Now hereby we have to study this genomics and proteomics means. So proteomics can be studied by using the knowledge of the genomics. Okay, proteomics means study of the proteins that can be studied by using the knowledge of the genome, because we know that genes code for mRNA encodes a protein. So here at a particular time, we know that at a particular time a gene is on in one particular cell is on, and it will be off in another cell. Okay, in particular time gene is on in a particular cell and it will be off in another cell. So here expression of the protein during the growth and development, that will be different from the resting cell, and even proteins produced by the GIT, gastrointestinal tract, and neurons will be different from the other proteins. And few proteins, they may undergo post-translational modification. All the proteins may not undergo post-translational modification. Post-translational modification means after translation is nothing but protein synthesis. After synthesis to become fully functional, few proteins may undergo post-translational modifications. So only few may be undergo post-translational modification. In same time, same gene will be off in one cell and that will be active in the other cell. And expression of the proteins during growth and development will be different with the normal resting cell. So here these are the difference. Now here but the genes are same in all the cell at all times. Okay, but they could perform different functions. Okay, but genes are same in all the cell at all times. So what human body has nearly hundreds different cells there, hundreds of different cells are there which express thousands of proteins at different times. Okay, so that's what this is the brief introduction about the genomics and proteomics. Now here the final lastly let us discuss about the determination of the protein structure. Okay, so how the protein structure is determined. So insulin was the first protein to be separated. Now here to study the already we have discussed this structure of the insulin and the primary structure of the protein. How the insulin is it is made up of two polypeptides, A chain and B chain, and how many amino acids, 51 amino acids are there. Okay, and 30 amino acids in one chain and 21 amino acids in another chain. So that is the insulin was the first protein to be separated. And in order to study the protein structure, first we should get the pure sample of the protein. Okay, see here now we are determining the protein structure. In order to determine a protein structure, first we should get the pure sample of the protein. How the pure sample is obtained means, so the proteins are extracted and purified by various chromatography techniques. By using chromatography is a technique by using that we can extract the proteins and purify. Examples of the chromatography techniques are like by using ion exchange chromatography, adsorption chromatography, HPLC, high performance liquid chromatography, partition chromatography. By using various chromatographic techniques, first proteins are extracted and purified. The purity can be checked by another technique, that is electrophoresis. That is electrophoresis may be agar electrophoresis, page electrophoresis, polyacrylamide gel electrophoresis. Page means or isoelectric focusing. Now the molecular weight is determined by mass spectrophotometer. Okay, first here in order to study the protein structure, first pure sample has to be obtained by various chromatography technique. Okay, and the purity can be checked by different electrophoretic processes, and the molecular weight is determined by mass spectrophotometer. Now determination of the primary structure of a protein. Okay, primary structure means we know that it's a linear sequence of the amino acids joined by a peptide bond. So how to determine a primary structure of a protein? Okay, linear number and sequence of the amino acids. Primary structure denotes the sequence, number and sequence of the amino acids. How this will be determined? So there are steps are there to determine the primary structure of a protein. First step is to determine the number of polypeptide chain in a protein. Okay, already we know that few proteins are monomers and few proteins are oligomers. Monomers means it is having only one polypeptide chain, and oligomeric proteins means they may contain more than one polypeptide chain. So first we have to determine how many number of polypeptide chains are there in a protein. So how to determine means, first the isolated and purified protein. Already we have isolated the protein and purified the protein by the techniques chromatography and electrophoresis. Now the isolated and purified protein is dissociated into subunits by treating with the HCl or the urea. Okay, the separated, extracted, isolated and purified protein now that is subjected to HCl, that is treated with the concentrated HCl or urea. That treating of the protein with the concentrated HCl or urea that may disrupt the non-covalent bonds and dissociates into polypeptide chains. Non-covalent bonds can be cleaved by treating the protein with the HCl and urea. Then by disrupting the non-covalent bonds, this proteins is separated into polypeptides. Even the disulfide bonds are also there in the primary structure that are removed by treating with the beta-mercaptoethanol. Okay, now here we are determining the how many number of polypeptide chains in a protein. So that protein first, the purified protein is first it is treated with the HCl or urea that dissociates the protein into its subunits. Subunit is nothing but each polypeptide is, you can call it as a monomer or subunit. So first protein is dissociated into subunits by treating with the HCl or urea. Treating of this with the HCl or urea may break the non-covalent bonds and dissociates into polypeptide chains. Even the disulfide bonds are removed by treating with the beta-mercaptoethanol. Now the number of polypeptide chains can be identified by treating with the diacetyl chloride. Now disulfide bonds have been broken down by treating with the mercaptoethanol. Non-covalent bonds has been hydrolyzed by treating with the HCl and urea. Now the protein, okay, the protein is dissociated into polypeptide, means a single polypeptide chain. That polypeptide chain, how many number of polypeptide chains are there, that can be identified by treating with the diacetyl chloride. So diacetyl chloride is a special, it's a chemical. It specifically binds with the only N-terminal amino acid of a polypeptide to form N-terminal diacetyl chloride. If you remember the structure of a protein, primary structure of a protein, what is the sequence of the amino acids are there? Amino acids are joined by peptide bond, and even the disulfide bonds are there, which connects the sulfidal groups of cysteine. Now here, if in the linear sequence of the amino acid, always there will be free N-terminal, where the free NH2 group is there on the left side, that you can call it as N-terminal. On either side of the polypeptide chain, always the carboxyl group is free. That end you can call it as C-terminal. Now here, the number of polypeptide chains can be, how many number of polypeptide chains are there in that protein, can be determined by treating with the diacetyl chloride. So diacetyl chloride, it specifically binds to the N-terminal amino acid of a polypeptide to form, okay, N-terminal amino acid react with N-terminal amino acids to form N-terminal diacetyl chloride. Number of diacetyl amino acids formed determine the number of polypeptide subunits in a protein. Okay, it means one. If two are there, if two N-terminal diacetyl amino acids are formed, that indicates that protein contains two polypeptide chains, like that. If four are there, if four N-terminal diacetyl amino acids are formed, that indicates the given protein contain like four polypeptide chains, okay, like that. So the number of diacetyl amino acids formed determine the number of polypeptide subunits in the protein. Now the tagged polypeptide chains tagged with the diacetyl chloride. Okay, now the tagged polypeptide chains are subjected to acids or enzymatic hydrolysis by boiling with the HCl at 110 degrees centigrade for 18 to 36 hours under anaerobic conditions, gives a mixture of amino acids. Okay, by treating with the diacetyl chloride specifically binds to the N-terminal amino acid. You will be knowing the how many peptide bonds are there in that protein. Now, the individual polypeptide chains are now treated with the enzyme. Enzymatic can be hydrolyzed by using the enzymes or by using the acid like concentrated HCl, boiling the polypeptide chain with the acid HCl for at 110 degrees centigrade for 18 to 36 hours. That can that can hydrolyze the polypeptide chains into mixture of amino acids. Now the amino acid composition is determined by chromatography. So number and nature of dancil chloride, dancil amino acid indicate the number of polypeptide chains. If two different polypeptide chains in a protein, two different dancil amino acids will be identified. Okay, this is the determination of primary structure of a protein. So what are the steps involved in the determination of primary structure of amino acid? First step is first we should know how many polypeptide chains are there. So to know that one, first we have to treat that protein with the HCl or urea that disturb the non-covalent bonds and converts breaks the polypeptide chain and that will dissociate the protein into subunits, and disulfide bonds can be removed by treating with the beta mercaptoethanol. Now the number of polypeptide chains can be identified by treating with the dancil chloride. We know that dancil chloride is specific in binding with the N-terminal amino acid to form a tagged polypeptide, that is N-terminal dancil amino acid. Depending upon the number of the dancil amino acids formed, that indicates the number of polypeptide chains are there in the protein. If two different polypeptide chains in a protein are there, if the protein has two polypeptide chains, we know that two different dancil amino acids will be identified. Once the tagged polypeptide chain, once the number of polypeptide chains has been identified, then it is subjected to acid by boiling at 110 degrees centigrade with the acid HCl for nearly 18 to 36 hours. That will be dissociated into mixture of amino acids. That amino acids then the amino acid composition can be determined by chromatography. Okay, so that's it about this one. And the next one is the determination of amino acid sequence of the isolated monomeric protein. Now we know that only by using the previous method, we have isolated the monomeric protein, each single individual polypeptide chains. But now we are determining the amino acid sequence in the given protein. How to determine? We know that each protein has a unique sequence of the amino acid. So to determine the amino acid sequence, there are different methods are there, different steps are there to determine the amino acid sequence of the isolated monomeric protein. Monomeric protein means it's a single polypeptide chain. So the steps in determining the sequence and number of this amino acid of a protein are, there are different steps, stepwise manner we have to go to identify the amino acid sequence. First we have to determine the N-terminal amino acid and the determination of C-terminal amino acid, determination of number and sequence of the amino acids. So stepwise manner we have to go. N-terminal amino acid we have to determine, C-terminal amino acid we have to determine, and then we have to determine the number and sequence of the amino acid. Finally, the protein is fragmented, hydrolyzed by using the digestive endopeptidases like trypsin, chymotrypsin, cyanogen bromide into individual amino acids. Now here, the fragmented derivatives, they are subjected to sequencing by the following. Okay, amino acid sequencing can be done by the methods Sanger's method, Edman's method, and Dancil method. Now here, these three, please note down, these three are the methods which will determine the N-terminal amino acid. Now here, first step is the determination of N-terminal amino acid. N-terminal amino acid can be determined by various methods, that is Sanger's method, Edman's method, and Dancil method. Now here, what is Sanger's method? Sanger's reagent is fluorodinitrobenzene, FDNB. That is fluorodinitrobenzene is the Sanger's reagent. This Sanger's reagent is made to react with the N-terminal amino. It binds with the N-terminal amino acid to form dinitrophenol derivative of the peptide. Okay, now here protein is mixed with the Sanger's reagent, which is composed of fluorodinitrobenzene. Now the Sanger's reagent is it binds to the N-terminal amino acid to form dinitrophenyl derivative of the peptide. Now this derivative, dinitrophenyl derivative of the peptide is then hydrolyzed, separated by chromatography. Then the N-terminal amino acid is identified. Okay, like this Sanger's method, which will identify the N-terminal amino acid. So here you just remember the Sanger's reagent. That is Sanger's reagent is fluorodinitrobenzene. So this Sanger reagent is reactive with the N-terminal amino acid to form dinitrophenyl derivative of the peptide, and this one is subjected to is further hydrolyzed and separated by chromatography. Then the N-terminal amino acid is identified. Then the Edmann's method. This is the one more method to identify the N-terminal amino acid. Here the Edmann's reagent is phenyl isothiocyanate, and this phenyl isothiocyanate react with the N-terminal amino acid of a protein to form phenyl thiohydranthene derivative of the amino acid, and this derivative is then hydrolyzed, separated by chromatography. Then the N-terminal amino acid is identified. Chromatography. Which chromatography? High performance liquid chromatography. Now, okay, so then now the second amino acid becomes the N-terminal, which reacts with the Edmann's reagent and cycle continues. I think you are following. So this, by using this method, each time in each cycle, one amino acid is removed from the N-terminal. Okay, so like that we can determine the sequence of the amino acid. Okay, first Edmann's reagent with the N-terminal, and then one amino acid is removed from the N-terminal. Again, now the second one will become the first one, that will react with the Edmann's reagent and then remove the second one. After removing this, third one will become the first one. Like that, the cycle continues. Each time one amino acid from the N-terminal can be removed. Next one is diacetyl chloride method. Okay, this is the another method to determine the N-terminal amino acid. Okay, to determine the N-terminal amino acids, there are three methods are there: Sanger's method, Edmann's method, and diacetyl chloride method. Okay, diacetyl chloride is the its full form. It is abbreviated. Diacetyl stands for dimethyl amino naphthalene 5-sulfonyl chloride. Diacetyl is which represents dimethyl amino naphthalene 5-sulfonyl chloride. Now this diacetyl chloride, it reacts with the N-terminal. It binds with the N-terminal amino acid to form a fluorescent derivative of diacetyl chloride. Okay, this diacetyl compound, it's a chemical. It binds with the N-terminal amino acid to form fluorescent derivative of the diacetyl chloride. This derivative then further hydrolyzed, separated by chromatography, and then the N-terminal amino acid is determined. Now coming to the determination of C-terminal amino acid. Which amino acid is there in the C-terminal? How to determine means C-terminal amino acids can be determined by using the enzyme carboxypeptidase. Okay, carboxypeptidase is an enzyme. It is specific in breaking the peptide bond from the C-terminal. Okay, see here. The C-terminal amino acid can be determined by using the enzyme carboxypeptidase. This enzyme specific in hydrolyzing breaking the peptide bond from the C-terminal amino acid. Continuous action of this enzyme sequentially releases C-terminal amino acid. Okay, see here, this enzyme first break the amino acid peptide bond from the C-terminal and remove one amino acid from the C-terminal. Then the second one will become the C-terminal. Again, the continuous action of this carboxypeptidase, again it breaks the peptide bond from the C-terminal and remove that amino acid. So like that, continuous action of this enzyme sequentially releases C-terminal amino acids. Now released amino acids are then separated and identified by chromatography. Like that, this is the method involved. So these are the steps involved in the determination of C-terminal amino acid. And finally, determination of number and sequence of amino acids. Okay, till now we have discussed about the number of determination of N-terminal amino acid and determination of C-terminal amino acid. Now we are going to discuss about the determination of number and sequence of the amino acid. How many amino acids are there in the protein and their sequence. So finally the protein is subjected to second round of hydrolysis by using the different endopeptidases. Okay. Now finally the protein is subjected to the protein is hydrolyzed by using the various endopeptidases. So and chemical methods which produces overlapping set of peptides. So these fragments, even it can be treated with the chemicals that can produce the peptides. Endopeptidases will break the internal peptide bonds and convert the proteins into small fragments, and even the chemical methods also produces the small peptides. So these fragments are then separated and sequenced. Overlapping peptides are useful in the elucidation of sequence of amino acids of the protein. So in the chemical methods produces overlapping set of peptides which are useful in the elucidation of sequence of the amino acids in a protein. Now here, these are the methods involved in the amino acid sequencing, using the methods involved in the determination of N-terminal like Sanger, Sedmans and Dancil chloride, and determination of C-terminal amino acid, and the determination of number and sequence of amino acids. Now finally, the latest techniques which are used to determine the amino acid sequence are automated sequencing, X-ray crystallography, UV light spectroscopy, and NMR, that is nuclear magnetic resonance. Okay, the nuclear magnetic resonance, or these are the latest techniques which are used to determine the amino acid sequence. Okay, so that's it about the protein and amino acid and the protein chemistry. So here I think I've covered almost all the topics competencies. Okay, then we'll move on to the next one, that is lipid chemistry. Okay, give me 1 minute, I'll check the slides. See here, now I am going to start another chapter, that is chemistry of lipids. Okay, see here in this chapter, the objectives you will come to know is what are lipids, definition of a lipid, and the functions of the lipids, then classification of the lipids, how the lipids are classified, and classification in detail, and here properties of the lipids. So these are the objectives you should here we will discuss here under this chemistry of the lipids. And here what are lipids? We know that lipids are group of heterogeneous compounds, okay, organic group of heterogeneous compounds generally found in all living organisms, and these are biologically essential. Okay, lipids are biologically essential. These are group of heterogeneous compounds, organic compounds found in almost all the organisms, and here lipids are readily utilizable by the body, by the human beings, and they will perform various different functions. Okay, and if you see the definition of the lipids is, lipids are the compounds which are insoluble in water but soluble in non-polar organic solvents like benzene, ether, chloroform, etc. So the definition of lipids are group of heterogeneous compounds, and these are the organic compounds which are insoluble in water but soluble in non-polar organic solvents like benzene, ether, chloroform, etc. Then, what are the functions of the lipids? Biochemical functions of the lipids is mainly lipids are the storage forms of the energy. Okay, so they are called biological fluid. Metabolic fuels are the best sources of energy. Why they are called metabolic fuels means lipids on oxidation, means on utilization, they will yield 9 kilocalories of energy per 1 gram. 1 gram of lipid on oxidation, means oxidation is nothing but utilization, yields, generates 9 kilocalories of the energy. So they are the storage forms of the energy. And here mainly where the lipids are stored, they are stored in the adipose tissue, and the amount of the energy obtained from fat and oxidation is 9 kilocalories. So they are called metabolic fuels, or they are the best sources of energy. And the second function is they are the structural components of the biomembranes. Okay, if you see the components of the plasma membrane is phospholipids and cholesterol. These are the lipids which are the components of the biomembranes. And not only these lipids, as they are present on the biomembranes, they act as receptors, antigens, and they act as the membrane anchors of the protein. Okay, see here, lipids are the structural components of the biomembranes, and as they are present on the biomembranes, they act as receptors, antigens, and they may help in the binding of the protein. Now here, so these are the structural, the second important function is they are the structural components of the biomembranes. And the next one is insulation. See here, the subcutaneous fat which acts as thermal insulator. The subcutaneous fat which acts as thermal insulator that prevents the loss of the heat from the body and helps in maintaining the body temperature. Okay, third function is insulation. The subcutaneous fat which acts as a thermal insulator and that reduces the loss of the heat from the body and that contributing the maintenance of the body temperature. And the lipids which give shape to the counter to the body, and lipids they protect the internal organs. See here, by covering them and providing the cushioning effect. Lipids protects the internal organs by covering them and providing them a cushioning effect. And lipids are the metabolic regulators. They can regulate the metabolisms, various metabolisms, and they are the electric insulators in neurons. Okay, see here, these lipids are constitution, the nervous tissues are rich in lipids, especially sphingomyelins. Nervous tissues are rich in lipids. Example is sphingomyelin, which acts as insulator in myelin sheath. So, electric insulators in neurons, example is sphingomyelin. It's a lipid. And these lipids help in the absorption of the fat soluble, okay, absorption and utilization of fat soluble vitamins like A, D, E, K. Okay, so lipids help in the absorption of fat soluble vitamins A, D, E, K, and these are the lipids are the important dietary constituents and which improves the taste and palatability of the food, which makes the food palatable. Okay, and these are the main functions of the protein. In addition to that, even the cholesterol is a protein which serve as the precursor for the synthesis of the bile salts, steroid hormones, and vitamin D. Okay, other function of the lipid is cholesterol is one of the lipid. It serves as the precursor for the formation of the biological important compounds like bile salts. Okay, bile salts help in the digestion and absorption of the lipids, and even steroid hormones, they are derived, steroid hormones are also derived from cholesterol, and vitamin D, which plays an important role in calcium and phosphate metabolism, it is also derived from cholesterol. Okay, cholesterol being a lipid, and it serves as a precursor for synthesis of vitamin D, like steroid hormones, and then bile salts, and even these lipids in combination with the proteins. Okay, these are the lipoproteins. Okay, lipids in combination with the protein to form lipoproteins, and the lipoproteins are important cellular constituents, and they serve as they help in the transport of the lipids in the blood. Okay, see here, lipids in combination with the protein to form lipoproteins. They are the important constituents of the cellular cell, and which help in the transport of the lipids. Okay, these are the major functions of the lipids. Okay, they are the storage forms of energy. 1 gram of lipid on oxidation yield 9 kilocalories of energy. So they are the metabolic fuels, and they are the components of the cell membrane. Example is phospholipids and cholesterol. And as they are present on the plasma membrane, they act as receptors and antigens, and they maintain the body temperature. Subcutaneous fat prevents the heat loss from the body and maintains the body temperature, and they are involved in the formation of like surfactant. Few lipids, they may act as surfactants, emulsifiers, and help in the formation of soaps and detergents. And the next important, they are the important dietary constituents which improves the taste and palatability, and they protect the internal organs by providing the cushioning effect, and they give shape and counter to the body. And here the lipids are the electrical insulators in neurons, and the lipids help in the absorption of fat soluble vitamins like A, D, E, K. Here, and not only that, even the cholesterol, which serve as the precursor for the synthesis of the important biological compounds like vitamin D, bile acids, and bile salts, and steroid hormones. So these are the major functions of the lipids. Then coming to the clinical importance of the lipids. Okay, maybe if there is obesity, atherosclerosis, and cardiovascular diseases. In diabetes mellitus also there is deranged metabolism of lipids is seen. Fatty liver, liver storage disorders, and hypoproteinemias. So these are the clinical importance of the lipids. These are all associated with the lipid abnormalities, atherosclerosis, cardiovascular diseases, obesity. Even in the chronic diabetes mellitus also, lipid metabolism is altered. Accumulation of the fat in the liver is fatty liver, and all these are associated with the clinical manifestations. We'll discuss later. And excess lipids are stored to form lipid storage disorders associated with the clinical manifestation, and even lipoproteinemias. These are the clinical importance of the lipids. Then coming to the classification of the lipids. So lipids are mainly, already we know the definition. The common property of all the lipids are they are insoluble in water, soluble in organic solvents like ether, chloroform, alcohol, etc. So the lipids which are available in the nature can be broadly classified into 3 major classes: simple lipids, compound lipids, and derived lipids. Lipids are mainly classified into simple lipids, compound lipids, and derived lipids. Simple lipids examples are fats, oils, waxes. And compound lipids, okay, they see here derived lipids. Now here, what do you mean by simple lipids? Simple lipids means these are the ester. These are made up of only fatty acid and alcohol. Examples are fats, oils, and waxes. Compound lipids means in addition to the fatty acid and alcohol, they may contain the prosthetic groups. Additional groups are present along with the fatty acid and the alcohol. And compound lipids are of different examples are phospholipids, glycolipids, sulfolipids. And the third class is derived lipids. These are derived either from simple lipids or from compound lipids by hydrolysis. Examples are fatty acids, prostaglandins, steroids, etc. And there is other group of the miscellaneous lipids are there, which are carotenoids and squalene. Okay, see here. That is a brief classification about the lipids. So lipids are classified into simple lipids, compound lipids, and derived lipids. Simple lipids, why they are called simple lipid means they are composed of only fatty acid, free fatty acid, and alcohol. Only alcohol and fatty acid is there, free fatty acid is there. They are called simple lipids, and simple lipids are of 2 types, neutral fats and waxes. And the next one is compound lipids. What do you mean by compound lipids? Why compound means here, in addition to the fatty acid, and alcohol, they may contain additional additional groups. Those additional groups you can call it as prosthetic groups. Additional groups are present in compound lipids along with the free fatty acid and the glycerol. Examples of the compound lipids are phospholipids. Phospholipids means they are composed of free fatty acid, alcohol. Along with that, they may contain phosphoric acid. Phosphate groups are present and the nitrogen base is there. Okay, phospholipids are composed of fatty acid, alcohol. Along with that, they may contain a nitrogen base and phosphate. And glycolipids means here lipids in combination with the carbohydrate. Here the prosthetic group is carbohydrate. So carbohydrate in combination with the lipids to form glycolipids, which are the examples of compound lipids. Sulfolipids. Sulfolipids means lipids in these are the sulfur containing lipids. Okay, all these are the examples of the compound lipids. The next one is the third class is the derived lipids. Derived lipids means these are, the name indicates, derived either from simple lipid or from compound lipids. So the examples of the derived lipids are fatty acids, steroids, all fat soluble vitamins, cholesterol, all these, even the cholesterol, all these that comes under derived lipids. Okay, now here that is the brief introduction about the classification of the amino acids. Details will be given in the afternoon's class. Afternoon there is a class, biochemistry class 2 to 3. So in detail we'll discuss the classification in afternoon's class. Thank you.